Welcome back, students. Today we are going to discuss part 11 of our unit 2, that is cell as a unit of life. And before I start the main lecture, I would like to inform that the images used in these slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathi@iitk.ac.in and we shall remove the images. In this particular video lecture, we are going to discuss about cellular respiration, fermentation, and biological oxidation. First of all, let's understand the term cellular respiration. So, cellular respiration is a set of metabolic reaction and processes that takes place in the cell of organism and convert chemical energy from nutrient into ATP. Okay, so can I write down over here? Chemical energy to ATP. There are so many metabolic steps, metabolic reactions involved in this process, and this whole process is known as cellular respiration. Cellular respiration. Okay, so cellular respiration is a process by which biological fuel are oxidized in the presence of inorganic electron acceptors such as oxygen to drive the bulk production of ATP, and this ATP of course contain energy. Okay, so this is a process. Cellular respiration is a process by which biological fuel are oxidized in the presence of inorganic electron acceptor, means oxygen, to drive the bulk production of ATP, right? And organisms that do not depend upon the oxygen, they actually degrade their food stuff through a process known as fermentation. Okay, there are two type of organisms, aerobic and anaerobic, right? So anaerobic respiration occur in the absence of oxygen, while aerobic then require oxygen for their cellular respiration purpose. Clear? Now, there are so many metabolic steps are involved. The first step is known as glycolysis. Let's understand the term first. Glyco plus lysis. Glyco means sugar. Glycose means sugar. And lysis means splitting. So, splitting of sugar occur in this particular process, right? And this scheme was first given by three very renowned scientists, Embden, Meyerhoff, and J. Parhans. So that's why we can also use the term EMP pathway for the same. So can I write down over here glycolysis or EMP pathway? EMP pathway, right? So in anaerobic organisms, this is the only process of respiration, because glycol in this particular step, glycolysis doesn't need oxygen. Okay, so in anaerobic organisms, this is the only process of respiration, and this glycolysis occur in the cytoplasm. So in certain prokaryotic cells, prokaryotic cell doesn't have membrane bound organelle, means they don't have mitochondria. Okay, so in the absence of mitochondria also they require energy for their metabolic steps, metabolism, which continuously going on in their cell for their development purpose, for their reproduction purpose, and for all the life processes they also require energy. If they don't have mitochondria, means they don't generate energy? No. Through glycolysis they can generate energy, because this glycolysis occur only and only in cytoplasm of the cell. And cytoplasm and cell membrane represent all type of cell, whether it is prokaryote, eukaryote, or whatever. And that's why we can use the term universal pathway for the same. So can I write down over here? Universal pathway. So glycolysis is also known as EMP pathway. It is also known as universal pathway because glycolysis occur in cytoplasm, and cytoplasm is present in all type of cells. Okay, and in this glycolysis, glucose actually undergo partial oxidation. Partial oxidation of glucose occur, and glucose is then further convert or split into two molecule of pyruvic acid. So this is glucose, which is a 6 carbon molecule. Through various steps, mainly 10 steps are there, it will convert into two molecule of 3 carbon molecule, 3 carbon, that is pyruvic acid. So partial oxidation of glucose occur in glycolysis. We are using the term partial oxidation continuously because one molecule of glucose, that is 6 carbon molecule, is convert into two pyruvic acid, that is 3 carbon molecule. So only single splitting of or splitting of one molecule of glucose into two molecule, only one this 1 2 3 4 5 6, only splitting or breakdown of one molecule occur, one bond occur, sorry, one bond carbon carbon, one bond, this breakdown occur. That's why we can use the term partial oxidation. If breakdown of all the bonds occur, that time we can use the term complete oxidation, and complete oxidation require mitochondria because other pathways after the glycolysis like Krebs cycle, oxidate and EMP pathway, that require mitochondria. Okay, clear? So that's why we can use the term partial oxidation. Partial oxidation of glucose occur in glycolysis. Clear? So in glycolysis, first this sucrose, which is a disaccharide, which is also known as invert sugar, it is converted into its monomeric unit or monosaccharide, or we can say breakdown of a disaccharide, that is sucrose, into monosaccharide, that is glucose and fructose, occur in glycolysis, right? And this in the presence of invertase. Why? Because we can use the term invert sugar for sucrose. Sucrose is also known as invert sugar. So invertase is actually the enzyme which involve the breakdown of this sucrose into its monosaccharide unit, that is sucrose and fructose. And these monosaccharide further enter into the glycolysis pathway. Okay, these two monosaccharide readily enter in the glycolytic pathway. It is mentioned over here. Okay, so this glucose and sucrose are phosphorylated to give rise glucose 6-phosphate and fructose 6-phosphate, we can say. Okay, as glucose is a 6 carbon molecule, so that's why we can use the term hexa. Kinase means ATP is involved, so kinase. So this glucose is convert into glucose 6-phosphate in the presence of ATP, because ATP give its one phosphorus molecule to glucose, and this phosphorus just attached to the 6 carbon sugar. That's why it is written over here glucose 6-phosphate in the presence of hexokinase enzyme. Why? Hexa for glucose, kinase means ATP is utilizing over here. ATP give its 1-phosphate molecule to glucose, and then glucose then further convert into fructose. It's isomer fructose, so glucose 6-phosphate to fructose 6-phosphate. Further, this fructose again accept another phosphorus from ATP, and this another phosphorus attached its first carbon of fructose. That's why it is written over here fructose 6-phosphate to fructose 1,6-phosphate. Okay, and then fructose 1,6-phosphate then convert into 2 molecule of 3 carbon, that is glyceraldehyde 3-phosphate. One is aldehyde, another is ketone dihydroxyacetone phosphate. This is 3 carbon and this one is again 3 carbon. Okay, and then this glyceraldehyde 3-phosphate, 2 molecules, they can convert to each other now, so that's why it is written over here. We can write down 2 into next type of molecule, means triose biphosphate. Okay, so 1,3-bisphosphoglyceric acid. So there are so many steps of this glycolysis. Once again, I'll repeat all the steps. In first step, glucose is converted into glucose 6-phosphate. And this phosphate is utilized by the glucose from where? From ATP. ATP give its phosphorus to glucose and convert into ADP. So glucose to glucose 6-phosphate, and then glucose 6-phosphate to fructose 6-phosphate, and further fructose again utilize 1-phosphate and then convert itself into fructose 1,6-bisphosphate. Here also one ATP has been utilized, another ATP has been utilized in this step. So first we utilize ATP for the production of further ATP. Initially we have invested ATP. Okay, then only a small amount of ATP we have invested in this pathway, and then after that we have generate ATP during the whole cycle. Okay, so glucose to glucose 6-phosphate, then glucose 6-phosphate to fructose 6-phosphate, then fructose 6-phosphate to fructose 1,6-bisphosphate, and then fructose 1,6-bisphosphate convert into 3 carbon molecule, 2 molecules of 3 carbon, that is glyceraldehyde 3-phosphate, and DHAP, that is dihydroxyacetone phosphate. Okay, and then this glyceraldehyde 3-phosphate then convert into 1,3-biphosphoglyceric acid, and at this time again oxidation occur, means it utilize the hydrogen atom of the utilize the hydrogen atom present in the system, and then this 3-phosphoglyceric acid convert into 2-phosphoglyceric acid, means this position of this phosphorus has been changed from third position to second carbon, to second carbon, and then 2-phosphoglycerate convert into phosphoenol pyruvate, and then phosphoenol pyruvate converted into pyruvic acid. So from 1 molecule of glucose, 2 molecule of 3 carbon pyruvic acid has been generated, and in this process 1 ATP has been produced, and in this step 1 ATP has been produced, and this is for 2, means 2 ATP has been produced on this step, 2 ATP has been produced on this step, means in initial phase we have utilized 2 ATP, and in the last steps we have generated 4 ATP, right, and 1 NADP, right. So here we can say 4 ATP generated while we have utilized 2 ATP. So total gain 2 ATP. In this, not total, net gain. We can use the term net gain, net profit, 2 ATP in this glycolysis pathway. While one molecule of NADH with one molecule of glyceraldehyde 3-phosphate, and here two molecules of 3 carbon sugar means 2 NADPH, 2 NADH. Okay, so we can say 2 ATP and 2 molecule of NADH, and this NADH then convert into ATP in mitochondria. Okay, so it will not we cannot say that we have gained this much of ATP, as NADH is equivalent to 3 ATP molecule, but we cannot convert that NADH in over here in glycolysis pathway. It need a mitochondria for the conversion purpose, because this NADH and FADH need a kind of bank for the conversion itself into ATP molecule, right? So in glycolysis pathway, this is the net profit. Net profit 2 ATP, because initially we have invested 2 ATP and total 4 ATP we have generated, right? And 2 molecule of NADH we have generated, and this one is convert into ATP molecule in mitochondria in other pathway. It require mitochondria, and this glycolysis occur only in the cytoplasm. So here only 2 ATP is our net profit. And what is the last product of glycolysis? This is pyruvic acid. So pyruvic acid is the key product of glycolysis. Now question arises, what is the metabolic fate of pyruvate? It actually depends upon the cellular need, whether next type of reaction is in the presence of oxygen or in the absence of oxygen. So it depends upon the cellular need, what kind or in which pathway this pyruvate has been followed, right? Let's say there are 3 major ways to handle this pyruvic acid which is produced by glycolysis. One is lactic acid fermentation, second is alcoholic fermentation, and third is aerobic respiration. Okay, this fermentation takes place under anaerobic conditions, means in the absence of oxygen, in many prokaryotic cells and many eukaryotic unicellular eukaryotes as well. Okay, and for the complete oxidation of glucose into carbon dioxide and H2O, it require, that particular organism require to adopt Krebs cycle, and this Krebs cycle need mitochondria, right? And which we also use the term aerobic respiration for the same, and that aerobic respiration require oxygen. So in anaerobic condition or anaerobic cells and or anaerobic organisms like prokaryotes, some unicellular organism, fermentation occur. Okay, it may be lactic acid fermentation or it may be alcoholic fermentation. So this pyruvic acid can follow this pathway, it can follow this pathway, or it can follow this pathway. It depends upon the presence of oxygen or absence of oxygen. Clear? Now, fermentation is also known as anaerobic respiration, and fermentation is the breaking down of sugar molecule into simpler compound to produce substance that can be used in making chemical energy. This fermentation process was first discovered by Louis Pasteur, And we can also use the term zymology for this science, fermentation. Science of fermentation is also known as zymology. And fermentation is also known as incomplete oxidation of glucose, because during fermentation total bond present in the carbon carbon bond, pyruvic acid, it will not total splitting or breakdown of all the bonds which are present in pyruvic acid. Pyruvic acid is a 3 carbon compound. Breakage of all carbon bond doesn't occur in this process. That's why we can use the term incomplete oxidation of glucose, because under anaerobic condition incomplete oxidation occur in fermentation. Okay, so fermentation occur in bacteria, anaerobic bacteria, certain kind of yeast, Saccharomyces, and muscle cells of animals. If blood supply is not that much vigorous, continuous, then that time also body cell require energy. Immediately cells switch from aerobic to anaerobic conditions and start to producing um, lactic acid. Okay, so in muscle cell also. So fermentation are two types: alcoholic fermentation and lactic acid fermentation. Some lactic acid bacteria utilize the same process. Some yeast, as I told you, yeast is used in the production of alcohol. Yeast is used in the bakery, different products of bakery also. People utilize this process, right? And some fungus also, right? So first of all, we right now we are discussing about alcohol fermentation. As you can see in this particular pathway, these are the steps of glycolysis. Glucose first convert into glucose 3-phosphate, then fructose 3-phosphate, then fructose 1,6-phosphate, then 3-phosphoglyceric acid, then phosphoenol pyruvate, and then this is the final product, pyruvic acid, which is a 3 carbon compound, right? And when this pyruvic acid gets converted into ethanol, that is 2 carbon compound, and it release 1 carbon dioxide, means decarboxylation occur, then this is known as alcohol fermentation. And this time pyruvic acid decarboxylase enzyme has been utilized. So can I write down over here pyruvic acid decarboxylation, decarboxylase enzyme. Decarboxylase means removal of 1 carbon dioxide. Decarboxylation means 3 carbon to 2 carbon molecule from pyruvic acid to ethanol occur during this process. Okay, so pyruvic or we can say alcohol dehydrogenase for the same reaction. This enzyme also involved in this particular step. So enzyme pyruvic acid decarboxylase and alcohol dehydrogenase catalyze this reaction, conversion of pyruvic acid to alcohol. Okay, this is known as alcohol fermentation, when the sugar or we can say sucrose utilizing all the steps of glycolysis and convert into ethanol, and at this particular point carbon dioxide has been released and some amount of energy also generated. What is the net ATP molecule produced during alcohol fermentation? Only 2 molecule of ATP. As just now we have discussed glycolysis pathway, 2 ATP net profit, 2 ATP. And in this process only NAD has been produced, there is no ATP formation. Okay, total 4 ATP has been produced from glycolysis, but 2 ATP has been utilized in first 2 steps just now we have discussed. So net gain 2 ATP. Okay, clear? So in alcohol fermentation, conversion of pyruvic acid into ethanol occur, carbon dioxide has been released, some energy also generated. Okay, and this alcohol fermentation, continuous generation of alcohol, if this process occur continuously, if the percentage of alcohol increase more than 13%, then the organism which actually involved in this conversion, that is yeast, this yeast poisoned themselves to death. This is so much harmful, this alcohol. If concentration of alcohol increase about 13% or more than 13%, that time yeast poison themselves to death when the concentration of alcohol reach about 13%. This is so harmful, this ethanol. Okay, but in the laboratory you have must have seen a 70% alcohol, 100% alcohol. That type of alcohol has been produced through distillation process only, and that is very toxic. Okay, yes. So this is about the alcohol fermentation. Next is lactic acid fermentation. When this pyruvic acid get convert into lactic acid bacteria are there, right? They are involved in this conversion. Some our muscle cells, when initially days, if we freshly join some sports classes, some gym, and some rigorous physical activity, we are involved in that. In initial days we feel so much muscle cramps in our body during fast running and other exercise. Why? Because in initial days, in our muscle cells doesn't in a very routine way, it doesn't require so much energy because we are lethargic that time. But immediately we start uh, activate them, that time blood supply is not enough, so that time that muscle cells are utilize anaerobic pathway for the production of their energy. So that's why it is written over here, during heavy exercise, some of our muscles respire anaerobically. And during anaerobic conversion, this pyruvic acid get converted into lactic acid, and that lactic acid is, showing its effect, means muscle cramps. We feel that time muscle cramps. Okay, so anaerobic respiration by muscles bring about partial breakdown of glucose to form lactic acid. Pyruvic acid in muscle cell to reduce lactic acid by lactate dehydrogenase. So, these are the steps of glycolysis. All the steps of glycolysis. When muscle cells get convert this pyruvic acid into lactic acid, that time we can use the term lactic acid fermentation. Okay, lactic acid fermentation. Here also this NADH get converted into NAD. Clear? And in this lactic acid, as I told you, lactic acid get accumulated in our muscles and cause muscle cramps. So at that time, our elder people and trainers suggested us to put our leg and that body part under a hot bath, right? So water bath give us some relief. Why? Because this hot water improve blood supply, blood flow. So oxygen supply increase slowly, and this oxygen start breaking down the lactic acid into carbon dioxide and water, and that's how it give us a relief from that muscle cramps. Clear? So this step, pyruvic acid to lactic acid, this conversion is known as lactic acid fermentation. Okay, so when glucose get converted into lactic acid in the absence of oxygen, when there is no oxygen in that type of cell, so in the presence of lactate dehydrogenase enzyme, our muscles convert this glucose into lactic acid and generate some energy which is required that time. okay, through lactic acid fermentation. ठीक है. So here also only net gain occur only 2 ATP. This is net gain. Net ATP production from fermentation of one glucose molecule to 2 ATP. Although total ATP production 4, but 2 we have already utilized in initial steps. Okay, so that's why it is written over here 2 ATP utilized. So net gain 2 ATP. Okay, clear? Now, fermentation limitations. In fermentation, very less energy has been produced, as just now we have seen only 2 ATP molecule from one glucose molecule. Very less energy is produced in anaerobic respiration. Less than 7% of energy in glucose is released, only 7%. Okay, not all of it is trapped as high energy bond of ATP because partial oxidation occur. ठीक है? And fermentation process is very hazardous because whether it is lactic acid or ethanol, that is hazardous. That is either acid or alcohol. These all are already we have discussed na 13%. If that particular limit reaches 13%, that the production organism, that is yeast itself kill itself. That is so much harmful. Now, biological oxidation. Energy is required to maintain the structure and function of living cells. True. Energy is driven from oxidation of carbohydrate, from lipid and protein. This one is again true. And the energy liberated is converted into ATP, which is known as energy currency of the living cell. And this oxidation process in living cell is called biological oxidation. So what is the definition? Oxidation process in biological system is called biological oxidation. So during this process, high energy compound is converted into low energy compound. So there is a liberation of energy occur in the form of heat, which is then further convert into chemical energy, means ATP. There are certain enzymes, certain groups of enzyme involved in this conversion, like oxidases, dehydrogenases, hydroperoxidases, and oxygenases. Okay, and some coenzymes are also involved in this biological oxidation, like NAD, NADP, FMN, FAT. These are certain coenzymes which involve in this biological oxidation. So first of all, let's understand oxidases enzyme group. These enzymes are actually responsible for catalyzing the elimination of hydrogen from the substrate. As this is a substrate, this oxidases enzyme involve the elimination of this hydrogen, and this hydrogen further accepted by this oxygen and produce water. There are certain examples of this group like cytochrome oxidases, L-amino acid oxidases, xanthine oxidases. So what is the main role of oxidases enzymes? Elimination of hydrogen from the substrate. Clear? Next group, dehydrogenases. This group of enzyme actually catalyze the removal of hydrogen from the substrate without utilizing oxygen or without using oxygen, means removal of this hydrogen occur, and this process is catalyzed by this enzyme dehydrogenases. So what is the final acceptor of this hydrogen? This is NAD or NADP. This is further accepted by whether either NAD or NADP. If reaction type is catabolic, means glycolysis, TCA cycle, then NAD act as a coenzyme. If reaction type is of a biosynthetic pathway, means fatty acid biosynthesis, cholesterol biosynthesis. That time NADP act as a coenzyme, because NADP are involved in anabolic reaction type and NADP involved in catabolic reaction type. Okay, so what is the main use of this dehydrogenases? Removal of hydrogen. It is mentioned over here, removal of hydrogen without using oxygen as hydrogen acceptor. Hydrogen acceptor present over here is not oxygen. This is either NAD or NADP. Clear? Next type, hydroperoxidases. Two type of enzymes are present in this particular group. These are found in animal and plant category, and these enzymes use hydrogen peroxide H2O2 as a substrate to form water. Peroxidases and catalases are the examples. Next, oxygenases. An oxygenase is any enzyme that oxidize a substrate by transferring the oxygen from molecular oxygen to it. Two type, monooxygenases, which transfer one oxygen atom to the substrate and reduce the other oxygen atom to water, and dioxygenases, which actually incorporate both atom of molecular oxygen into product of the reaction, or we can say oxygen transferases. Dioxygenases, what is the next term of this dioxygenases? Oxygen transferase. It incorporate both atom of oxygen, these two, into the product of the reaction. Okay, so this is all about the biological oxidation and glycolysis and fermentation. I hope all the concept has been clear now. Still you have any query, any doubt related to this topic, kindly write down your query in the comment box. We will discuss it later on. Till then, take care. Bye bye. Namaskar.